Hello, everyone. My name is Andrew Saidan, and I'm here with.
Anna Jones, hey y'all.
We're here to present crop evaluation on the greenhouse management class that we took this semester. So we will start with a table of content, and here we have production steps and activities that we're going to talk about in this little in a few minutes, and then we talk about the plants that we grew in the greenhouse, and then we will talk a little bit about the pest and disease that we identified, the fertilization process, the culture practices as well. Then we will also talk about the things that went well and those that didn't go on well, and then we end with our recommendations. So with the production steps and activities, we started with the irrigation setup systems. So here, the students together with the technician in the lab um, designed, set up the irrigation systems to help the plants. And then after the irrigation system were done, the next thing was the plug pots up. Here we introduced the plants that we had in the greenhouse into the pots, and then arrange them on um, the benches in the greenhouse in order of their names. And after that, we apply the fertilizer to the system, and with that, we did 300 ppm for the poinsettias, and then 150 ppm for the other plants all put together. Then after that, a couple of days, realized that the plants were growing very healthy, and then the poinsettia reached a point whereby we needed to start pinching to help it grow bushy instead of growing tall. And after that, realized that the plants were growing very well, and we needed to control the growth of the plant so that it doesn't get ready before the plant sales day. So we had to in, apply plant growth regulator to the plants in the greenhouse to regulate their growth. After that, students were put in groups to work on each bench, taking the pH and the EC of the plants that were in the greenhouse through the pour-through methods. And whilst we're taking the pH and the EC, we're also looking through the plants, trying to identify diseases and pests, and report that to um, the professor as well as the other team that will be taking over from us in the group. Then after that, we ended the class with the plant sales, and the plant sales was into October 20th, and then November 27th, which was the main plant sales day. Here I'll talk a little bit about the plants that we grew in the greenhouse. Um, about 7 plants, that is chrysanthemums, poinsettia, echinacea, gabardine, snapdragon, succulent, and poinsettia. So with the chrysanthemums, we had 2 colors, that was red and white, and with the cultivars, we had a jubilant red, touchdown white, they told that dark pink, and with the species was Morifolium. Then with the dianthus, there were 2 colors, the red and pink, and with the cultivars, there was strawberry, crimson eyes, crimson plated, and with the species was the chinsensus. And with the echinacea, it was just one color that was green. And with the cultivars, we had a green orange, scarlet white, and the species was um, Papyra. Then the next thing I'll talk about is the Garbaceae, and it was multiple colors. So with the cultivars too, we have strawberry, um, Cabernet, and then Altum. And with the species was Gaberia jamesoni. The next that I'll talk about is the Snapdragon, which also those 3 colors, that um, pink, red, and white, and with the cultivars were sun glow, scarlet, white, And then with the species was Anterin Anterinum negus. With the scarlet, succulents, which was last but one plant that I'll talk about. Also, we identified there was this one green color that we planted, and with the cultivars floor, and with the species was Gasteria. And the last plants, which is the Poinsettia, was also two colors, red and white. The cultivars were Freedom Red and Freedom White, and with the species was Ephobia poultryworm. Our the next thing I would like to talk about is the pests and diseases. And during our the greenhouse activities, we realized that there were these white flies and fungus gnats in the greenhouse. So we reported that to the greenhouse manager, and she, together with her teams, sprayed the greenhouse to control these pests that were identified. And then also fungicides were applied through the drenching process in the greenhouse to also help with the plant's growth. At this point, I hand over to Anne to continue on.
Hey, everybody. I'm going to start with fertilization. We use a brand called Jack's Professional Fertilizer with the ratio of 15:5:20. That fertilizer ran through a dosatron system, which was able to reach the plants via irrigation lines. So the poinsettias were set to receive a 300 ppm of nitrogen ratio, and all the other plants were set to receive the 150 ppm in. We also applied a PGR called Psychocell, and we did that to a drench or with a drench to the poinsettias at a, I think it was a 2,500 ppm ratio. Next, we have the cultural practices, and those are just referring to some of the things we did inside the greenhouse. And one thing that really stuck out to me was that the class was split into groups, rotating throughout each bench. Every week, the groups would get a different bench to take care of, and that really helped in making sure the plants were well taken care of. Another thing was good greenhouse etiquette. We had to make sure we hung the hose up and kept a tidy area in order to deter pests and diseases, and that it really helped. So moving on to what went good and what didn't. We noticed that the EC of the poinsettias was a little low, and we could have corrected that by increasing the injector ratio. There were also issues where the plants were not being watered properly because of the irrigation not being turned on. So they would turn off sometimes, by human error, and that could have been corrected just by knowing how to work the irrigation. But the irrigation did help us in keeping from watering the plants every day. So it was a big time saver there. Some recommendations that we came up with, and this was a big one, the plant cell timing. I feel if we were a little bit more engaged and put a little bit more thought into the timing of the sale, we could have sold a little bit more. And then the actual location, if we could have moved to a different, more trafficked area, I think it could have helped us a lot. And one thing that Andrew's come up with, and I agree with him fully, is that if we could have had a lecture or discussion on the marketing strategies for selling plants, that could have been a win for us. So yeah, that's all I have. And thank you guys for watching.
Thank you.